So, chapter 15 is blood stain analysis, and it has DNA in it. So, we'll do this and then we'll go back to drugs. Okay, so the first part of this chapter is about the nature of blood. So, what's actually in blood. So, blood has several different components. I have a picture on here, which it's a tube of blood, and it has plasma. Okay, so plasma is the liquid that's around the blood cells. So plasma is the liquid that's around the blood cells. They often describe it as straw colored, or it looks kind of yellow. Okay, the red blood cells are erythrocytes, so that's just another name for red blood cells. White blood cells are also called leukocytes, and there's not very many of them in your blood. And then there's platelets. Now, our red blood cells actually don't have DNA in them, but our white blood cells do. Okay, so plasma is the liquid, and then red blood cells, white blood cells, and platelets. And platelets are important for clotting. And we can talk about that more in lab if you want. Okay, now, how do we identify or type blood? So the kind of blood typing we do is called ABO typing. So that's based on the premise that on the surface of our red blood cells, okay, so we have red blood cells, and on the surface of them, there are antigens. Now, an antigen is just a substance that causes your body to have an immune response. And typically, that immune response is the idea that we make antibodies to them. And I'll try to explain all this in lab. Okay, so the antigens that are on our red blood cells are made out of protein and carbohydrates. And I have some pictures of them I can show you. So they're on the surface. And usually we just symbolize them as like A, B, and there's another one called RH. Okay, so A, B, and RH. So there are these things on our blood cells, and everybody has there are different groups of these antigens on our blood cells. So there's some that are A, there's some that are B, and then there's some that are RH. Okay, now, in our blood as well, we have antibodies. So we develop antibodies to the blood types that we don't have as part of our normal immune system. So when you're a baby, you start developing those antibodies. So we can type blood by having antibodies that match a specific antigen. Okay, so the fundamental principle of blood typing is that every antigen has a matching or like a corresponding antibody. Okay, so typically antibodies usually they're shaped like a Y, and they have two spots on them that are going to bind to the antigen. So what happens is you can have this Y, and the parts that bind are on the short arms of the Y. Okay, so the top part of the Y. They can bind to the antigens that are on the blood cells. So what happens is we can have a blood cell that has A on it, and then we'll have anti-A antibodies, and they'll bind to the blood cell. So we can buy these we can buy the antibodies. Okay, so we can purchase those to use them. We don't have to. We buy them to type blood. So we'll have anti-A antibodies. So they're going to bind to A-type blood. And when they bind, what it does is it causes all the blood cells to clump together. And the technical word for that clumping is called agglutination. And I'll show you in lab how this works so that you can see it. It'll make more sense, I think, if we talk about it together in lab and you can ask questions. And I'll draw some pictures on the board and try to explain it. Okay, so the picture that's in your book, it shows antibodies. They're to me, they almost look like a barbell. So they have like a Y on each end. So they have the A red blood cells with a circle on them, a semicircle, and that's supposed to represent the antigen. Okay, now anti-B antibodies will not bind with A because they're not the same shape. But anti-B antibodies will bind to the B antigen on the B blood cells. So I'll show you. And it causes them to clump together. We'll go over this in lab, so be in lab. And I'll try to find a video too to add. Okay, now there are some very complex antigen type blood typing systems. So there's more than 15 different systems that have been identified. So lots of times those are used when you're trying to find a match for a transplant. The most important ones that we use are ABO and RH. Okay, so if and I'll go through and I'll explain all this in lab how it works. The book kind of goes through it, but I think the book is kind of confusing. So we'll go over it in lab, and I have an activity where we can do it. We can test some fake blood. Okay, whoops, wait a minute, I gotta go back. Okay, now they have figured out the types of blood that exist in the overall population. So they say that approximately 43% of the population has type O blood. 42% has type A, 12% has type B, and 3% has type AB. Okay, there are some variations in that depending on ethnicity and where you're from. Okay, so we're going to go over blood typing in lab, and we're going to go over this RH factor, so whether you're positive or negative. We'll go over that in lab too. Okay, now, one thing that fits in with this chapter is these ideas of some of the tests that we actually do to look for drugs or look for something that could be present. Okay, so they're called immunoassays. So immuno is like immune system, and an assay is a test. So immunoassays are based on the idea that you could make an antibody for any type of antigen. Okay, so you could take anything that you wanted to, in theory, and you can make an antibody to it. Okay, so an example of two common immunoassays that you guys might be familiar with are a rapid strep test, okay, or a pregnancy test. So those are assays based on antibodies, okay, and antigens. So they take you have the strep antigen in you if you have strep throat, but they put the antibody on a strip, right? So then they'll swab your throat and they'll add the throat swab to the strip, and then that you could get a response. Okay, so immunoassays are used for urine drug screening. So whenever you have to go pee in a cup for a drug screen, they're going to use immunoassays for that. Okay, so if you can create an antibody for a drug, you could use it to test for the presence of the drug in body fluids. Okay, we don't normally have antibodies or an immune response to drugs that you take, especially like illicit drugs. However, you could take the drugs and you can combine them with a protein, and you can make a bigger molecule, and you could put that into an animal like a rabbit, and you could get the rabbit to make antibodies to the drug. This is a picture, and it's showing a drug. Okay, so we have a drug, and then we're going to bind it to a protein. Okay, so we hook it to a bigger protein carrier. Now the rabbit will have an immune response to that protein with the drug attached to it. So they put it into a rabbit, which it's a lab rabbit, so it hasn't been exposed to anything else. And there's a series of, there's like a protocol for how you put these into the rabbit to get good antibodies out. So you put it into the lab rabbit, and then you take the blood from some blood from the rabbit, okay, and in the plasma, that's where the antibodies are going to be. And you can purify the antibodies from that. So then you can get antibodies, and then you could use the antibodies for testing. And nowadays, you wouldn't have to do it with, you wouldn't have to make them with the rabbit. You could order them. You could just buy them. Okay, now, forensics of blood. So the first thing is you have to figure out, is it really blood? Is it blood? Is it ketchup? Is it, I don't know, spaghetti sauce? What is it? Is it blood? So, and then once you figure out, yes, we do have blood, then you have to figure out, where did it come from? A human? Did it come from an animal? Okay, then you have to figure out, the next thing would be, if the blood is from a human, could you associate it with a specific individual? Okay, so to figure out whether it's blood or not, there are some colorimetric or preliminary color tests. So the first test is called a Kastle-Meyer test. So what happens with it is you're looking for a pink color. So deep pink color is going to be the positive. So what happens is blood cells have peroxidase activity. So peroxidase is an enzyme. Okay, so if you take blood and you add hydrogen peroxide and you add phenolphthalein, which is a color indicator, if peroxidase is present, and we're saying blood is present, if peroxidase is present, then we'll get this pink color. They used to do it with another compound, but the other compound, not phenolphthalein, was actually a carcinogen. So then they started using phenolphthalein, which we use in the labs here because it's not bad for you and it's easy to see the color. Okay, now some people, some field agents, there are, if you're having your urine tested, like at the doctor's office, they just use a dipstick, and the dipstick can tell you whether you have blood in your urine or not. So some field agents might take some water and a cotton swab and pick up the blood and then put it on the urine dipstick test, and then if it turns positive, then it would indicate that that is blood. Okay, now the third test, this is more glamorous and sexy, is the luminol, or it's also called a blue star test. So luminol is super sensitive. So you spray it on. It's capable of detecting blood that's been diluted up to 100,000 times. So somebody's washed it. So you shine a UV light on it. So you have to turn the spray with the luminol. The luminol reacts with the blood, and then you shine the UV light on it, and you should be able to see a blue light. Okay, now the next thing, so you have blood, okay, and you want to figure out if it's coming from a human or from an animal. One of the tests to do this is called a precipitin test. Okay, so precipitin looks like precipitate, right? So that means something's going to come out of solution. Okay, so for this, what we're going to do is we're going to use antiserum. Okay, so antibodies, it's from rabbits, but the rabbits are being used to make antibodies to other animals. Okay, so like we can buy goat, or we can buy rabbit anti-goat. We could buy rabbit anti-deer antibody, rabbit anti-cow antibody. So we have antiserum that will react to blood from different species. I have a picture of it. Okay, so here's rabbit anti-human antibodies. So they took blood from a human, okay, and they put it into the rabbit. And then the rabbit starts making antibodies to the human blood. Okay, so then they take some serum from the rabbit. Okay, so they take the blood from the rabbit and collect the plasma and collect the serum. Okay, and then what they'll do is they'll put the rabbit serum in like a little tiny test tube. Okay, and then they're going to add the unknown blood to it. And they probably dilute it, so they'll take some of the, if it's a dried blood sample, they'll add a little bit of liquid water to it, right, and then suck it up, and then they'll layer that on top of the rabbit serum. And if there is a reaction, you'll get a precipitate, or like a little band of it that's cloudy at the interface of the human blood and the rabbit serum. So you'll see a reaction. It's not going to be like pink, but it'll be, you'll be able to see a change if there is a positive reaction. Okay, another way to do this instead of putting it in a tube is you can do it on an agar plate, and this is called gel diffusion. So the idea is you can take an agar plate, and I have a picture of it here. You can take an agar plate and have 2 holes in the agar plate, and you can add your unknown in one hole, one well, and you can add your antibody to the other one. And when they actually meet and they come together, they'll create a line. If they don't react, they don't create a line. I might have a lab we could actually do that with. It's kind of cool. Okay, so here's the picture from the book. So they're saying they have wells, and the wells are in a square agar sheet. Okay, and they added antigen to one. Okay, so that's the unknown. That's what we're trying to figure out. And they added antibody to the other. Then they added, they tried to speed it up. I've done this where you just let it sit and it just diffuses through. But what they did is they would want the results faster. So they add an electrical current to it, and the antigen and the antibody move towards each other. And if you get the line or a precipitate between the two, then it's a positive response. Okay, now to individualize blood stains, okay, to connect it to a single person, we're going to have to do DNA analysis, and we're going to talk about that later. Okay, now, testing for seminal stains. Okay, so there's a whole big section in here about how to look for semen. So in sex offenses, it's necessary to look for the presence of seminal stains. Okay, so we're looking for semen and sperm. So if you look at something and it's kind of like white and crusty, okay, then you can kind of assume that that is semen. Okay, but sometimes people will kind of wash it off or wipe it off. So then what you're going to need to do is more scientific testing. Okay, so the best way to locate and at the same time characterize a seminal stain is to perform this test. It's called acid phosphatase. So acid phosphatase is an enzyme that's found in seminal fluid. Okay, so it's actually part of the sperm. So acid phosphatase, if you add, I can't remember what the chemical is, but add a chemical to it, and if semen is present, the acid phosphatase will give you a purple color. Okay, so then another thing that you can do to confirm, there are other things that could give a positive for acid phosphatase. Okay, so there are some other things like vegetables and fruit that have acid phosphatase in them. So you need to keep looking. Okay, so one of the things that you could also look for is you could make a slide and look for sperm. Okay, then another variable that comes in is that not everyone who commits a sex crime makes sperm. So they could ejaculate and there might not be any sperm in there. It just might be semen. So you can also test for a protein. And the protein is goes by 2 different names. So P30 is one name, or prostate-specific antigen. So it started out as prostate-specific antigen, or PSA. Okay, but they found it in other tissue besides prostate, so now they try to call it P30. All right, so if you had a positive for the the acid phosphatase and a positive for this P30, then you could definitely say, yes, there's semen on it. Okay, now, we also in this chapter, they also talk about evidence from sexual assault. So the sexual assault victim should undergo a medical examination as soon as possible after the assault. Lots of times the hospitals have nurses that are specifically trained in this. They're like forensic nurses. So specific items are going to be collected, and there's going to be a kit, a sexual assault evidence kit. So they're going to look for pubic combings. They're going to take control pubic hair from the victim. They're going to do external genital swabs. They'll do vaginal swabs, cervix swabs, rectal swabs. They'll swab all different body areas that the suspect might have touched. They'll do oral swabs. Okay, then they're going to take some head hairs as control. They'll do a blood sample. They'll do a buccal swab for DNA, so that's cheek. They'll do fingernail scrapings. They'll also take a urine specimen, so they could get semen out of there. And they're going to try to sample all the clothing. So the clothing should be packaged separately. Okay, so it shouldn't all go in one bag. They should be in paper bags in case we need to get DNA evidence out of it. We want it to be dry. And the bedding or wherever the assault took place should also be collected or sampled. Okay, now, if the suspect's apprehended within 24 hours of the assault, it might be possible to find the victim's DNA on the male suspect's underwear, okay, or on the penile swab. So there could have been transfer to the guy's penis, and then it could have rubbed on his underwear. And there's a case study in the book. It talks about the fact that the woman had sex, like consensual sex, with one guy, and then she was raped. So they found her cells in the rapist's underwear, but they also found the consensual sex man's cells in the underwear. Okay, now, items that are going to be collected from the suspect, they're going to take the clothing, pubic hair, head hair, they're going to swab the penis, they're going to take a blood sample, they're going to take a buccal swab. They also might look for saliva because there's forceful contact, right? So they might look for more blood. They're going to look all over for semen. There might be fibers that have been exchanged. Okay, now, what they could still find in the vagina is some of the seminal constituents could still be in there for quite some time. Okay, so sperm that are still motile could be found in the vagina after 4 to 6 hours. Non-motile sperm could be found 3 to 6 days. Seminal acid phosphatase, it's going to be less than 48 hours. And then the prostate-specific antigen, the P30, is less than 72 hours. So it does persist in there for a little bit, but it's better to go right away. Okay, now, we've kind of talked about all this before. So packaging biological evidence. So it's important that everything's photographed and sketched before you start collecting the evidence. You're going to wear gloves. Don't put DNA evidence in something that's airtight. You don't want mold or moisture or bacteria to grow in there because it can break down the DNA. Any stained article should go into a separate bag or a well-ventilated box. And all biological evidence should be refrigerated or stored in a cool location until it gets to the lab. Okay, now I'm going to stop here and upload this because that's like 23 slides and I have about that many more to do.